Hey, Nikki, what are these beautiful pictures that we're looking at right here?
Yeah, these are actually photos of a skirball when we went to collect the soil after the fire recently in December.
Oh, so what were we doing there?
We are actually taking two different soil samples, one of a recovering soil area and one that had no recovery present.
Oh, wow, that's awesome. Are you like part of a research team, or how do you
Yeah,
guys go about this?
yeah, thanks for asking. I'm actually part of this really stellar research group through MIMG 109AL. And
Hello, everybody. My name is Jenny. I'm a Vietnamese American fourth year at UCLA, and I'm currently also an MIMG major and hopefully aiming to get into dental school. I'm also part of another research lab that explores the oral microbiome. So doing this lab is also pretty interesting as well. You discover two different microbiomes. But also in my free time, I like to play guitar or piano and paint and read.
Hello, my name is Ashley Choi. I'm a fourth year MIMG major at UCLA. I love drawing and painting, and I was once an art major. I'm a pre-med now, and I love microbiology, and I'm looking forward to learn the significance of microbiome in terms of medicine.
Hey guys, my name is Ian. I'm a third year MIMG student at UCLA, and I'm excited to be part of Nikki, Jenny, and Ashley's team. I grew up in Santa Clarita, California. It's about 30 minutes north of here. Fun fact, it gets up to 115 degrees in the summer. But one of my favorite hobbies is skiing and snowboarding. I've been going up to Mammoth since I was about 5, and I could do a 360.
hey guys, my name is Nikki Lambus. I'm currently a third year MIMG major here at UCLA. I'm actually from a small town in Arkansas called El Dorado. In my free time, I enjoy playing basketball and just hanging out. I'm really excited to finish our project in this lab and see where the results take us.
And I'm looking to develop a hypothesis regarding how fire affects the concentration of soil metabolites and bacterial taxa. What's the latest in the literature?
Surprisingly, plants and bacteria have well-documented symbiotic relationships, usually exchanging nutrients, and in many cases, plant health is linked to the microbial composition and nutrient availability. Specifically, nitrogen, phosphorus, potassium, iron, and carbon are essential for plant growth. And bacterial metabolic functions can change the concentration of these essential nutrients in the soils. For example, some bacteria can convert ammonia into usable nitrogen, while others can increase the availability of phosphate and iron. Surprisingly, major community shifting events such as wildfires have been documented to change the presence of free carbon, nitrogen, and these other minerals in the soil.
Hey Ian, you didn't mention how microbiome could be involved in regeneration of burned soil. I'm curious about how microbial properties can restore burned soil and eventually promote plant growth. So, we should ask ourselves, 1, what type of soil bacteria that survived from the wildfire would regenerate soil? And 2, what kind of other soil factors, such as mineral components, play a role in soil restoration after wildfire? I think we should perform functional and biochemical assays to measure metabolic activities and mineral composition. What do you think, Nikki?
Ian, since you mentioned that major community shifting events in soil can change the composition of the soil microbiome and the concentration of essential metabolic intermediates, you think if we cultivate isolates from fast and slow recovering soil, then we can conduct relevant biochemical and functional assays to deduce metabolic and mineralogical factors associated with soil regeneration?
So we'll be doing two types of experiments. The first one is a biochemical type of experiments, and it's basically to test the metabolic activity of the microbes that we extract from the soil. And the second one are functional experiments in which we kind of test, do they have properties that help regenerate the minerals inside the soil that help and promote plant growth. And then the last part of our experiment that we'll get to at the very end is something called metagenomic sequencing. That's basically looking at the genes of the different microbes and seeing where they match and how different they are. And that can give conclusions about the types of microbes that might be living inside the soil. And we can see if there's a difference between the soil where there's plant growth and the soil where there's not a lot of plant growth going on. So far results have shown that there really isn't too much of a difference, besides the fact that maybe the soil with low regeneration is particularly more diverse in the types of functions they might have, but overall they're pretty same amount of metabolic activities and functions so far. But we're still in the process of testing, so we'll see what results we'll get.
Our first question was, what do they do? Which involved functional assays and biochemical assays of isolates to learn their various metabolic properties. In order to answer our next question, once there, we will determine the identity of soil bacteria and their composition in the soil sample. First, we will look at metagenomic DNA from a soil sample to get a representation of the bacterial community. As a result, we can understand what kinds of bacteria live in the different conditions of soil and how the different composition between the non-recovering soil and recovering soil affect restoration in burned soil.
So our work has a lot of big picture resources. Surprisingly, the consumption of energy sources such as petroleum, oil, and coal by Western civilizations has been increasing every year, and if we keep consuming these resources at the current rate, the atmospheric CO2 concentration will increase by at least 50%. This phenomenon will only exacerbate global warming and makes finding an alternative energy source an immediate problem. One solution is the production of biofuels from energy crops such as corn, soybeans, and wheatgrass, which will need to be produced in massive quantities in order to replace current energy sources. Our research has the potential to further these efforts by identifying bacterial strains and metabolic properties associated with plant growth in order to provide insight into factors essential for soil revitalization. This information can also be used to identify high-yield crops to improve the composition of modern fertilizers, which would further increase crop yields for biofuel production.
As our research project has not yet concluded, our available results have yet to show a statistical significance between the two samples when taking into account the assays ran. However, there did seem to be more variation of responses in the non-recovering soil isolates. These inconsistencies do not support our hypothesis by showing that there seems to be deviation in the response of the isolates of the different soil samples.